welcome to WashU. And so Steve, did you? Okay, we're good. So welcome to WashU in the genetics program. We're certainly excited that you're here, and we want to thank you first for applying, and second for coming. We're going to welcome the international students too. Hopefully you get to see this on YouTube. We will see. I'm going to correct a few things on the schedule. Right now we're going to do the program overview. I think that was said to be after lunch, so we're going to do the program overview now. We'll talk about DDBS and the genetics program, primarily the genetics program. And then after lunch, it's going to be student groups and kind of a presentation on different student groups and special emphasis pathways. What are they? They're kind of like concentrations in college. So the first thing let's do is let's just do introductions. My name is Jim Skeet. You probably all got emails from me, and if you didn't, it's my fault, but I think I got all 15 emails. I'm in the genetics department. I've been here almost for 20 years now. Tim and I have run the genetics program 10 years. We think we're in our 10th year. It's either 10th or 11th year. It's been a while. Now, my research, I did my undergrad at Haverford College, a small college outside of Philly. I did graduate school at University of Wisconsin, and our research focuses on really nervous system development using flies as a model system.
So Tim Shadle, I'm also in the Department of Genetics. I was an undergrad at Lawrence University in Appleton, Wisconsin, and then went to University of Wisconsin in Madison. And I work on C. elegans genetics, understanding germ cells and germline stem cells.
I'm Albert Ville, for those of you who I haven't introduced myself to. I'm finishing my undergrad education at Gettysburg College this semester. And my research to this point has dealt with phage, genomics, bioinformatics, and ecology.
My name is Tomas Lagunas. I graduated from UC Riverside back in 2012. And for the past 3.5 years, I've been working in agricultural biotech.
I'm Jordan Brown. I'm finishing my undergrad at Marquette in Milwaukee, Wisconsin. And my research is looking at how microRNA function to control C. elegans ovulation.
Hi, my name is Nina Berry. I'm from the University of Maryland, Baltimore County, and right now I'm a lab tech working on degradation of plant matter by bacteria.
Hi, I'm Jenny Hazen. I'm completing my undergrad at The Ohio State University, and right now I'm working on novel RNA editing enzymes and RNA editing pathways.
I'm Penny. I'm studying at the University of Chicago. I work on yeast recombination.
My name is Ariel Vermuni. I just graduated from UNLV in December. My research basically used a bioinformatics approach to do more efficient transcript assembly and find novel promoter elements in rice.
I'm Matt Durant. I go to Brigham Young University. I'm a senior, studying neuroscience and bioinformatics, and currently researching common Y chromosome variants associated with phenotypes extracted from electronic health records.
I'm Brianna Middleman. I am finishing up undergrad at Duke University, and I study in a evolutionary genetics lab, and I study the genetic architecture of a sexually selected tree.
Hi, my name is Rachel Newcomb. I am a senior at the University of Kentucky, and my research experience has primarily been in the genetic determinants of drug sensitivity and cancer.
I'm Cynthia Tang. I graduated from the College of Idaho, and I'm now working as a postdoc here on long non-chronic RNAs and cancer.
I'm Justin Ronchioli. Did my undergrad at the University of Rochester, and I now work at the University of Pittsburgh, and I study the alternate lengthening of telomeres cancer pathway.
Hi, my name is Morgan Brissy. I graduated from University of Michigan in 2013. I'm currently a lab assistant in tech at Wayne State University in Detroit, and I primarily study the role of estrogen receptors in the onset of labor and preterm labor.
I'm Brianna B. Bell. I go to St. Mary's College in California, and I'm currently researching metallopeptidase biochemistry, so very different from
I'm Evan. I'm an undergrad here, and I have been researching kind of functional metagenomics and its applications to biofuel production.
Okay, thank you. Welcome, welcome. Okay. So over the next 45 minutes or so, we really want to do 5 things. We want to give you our perspective of these weekends, a little bit what we think your perspective of the weekend should be. Give kind of a relatively condensed overview of the structure of DBBS and the genetics program. So I don't know if you've probably heard or read that you have the DBBS, which is the umbrella program. There are 12 programs within it. What they don't tell you is there's kind of 11 programs, and the best program is genetics, because all of you applied for it. We're going to talk briefly about keys, what we think are 2 keys to success in graduate school. Talk about our philosophy of graduate education. We'll kind of finish up with Tim talking primarily about the first year kind of educational elements. As I mentioned, Evan, I mentioned this earlier, grad school is primarily about research, and at the same time the program focuses a lot on the early years, and the early years have both kind of didactic and clear core training elements and also research focus. So that's kind of what we're going to do over the next, hopefully, 45 minutes. So what's our view of the recruiting weekends, or interview weekends? I call them recruiting weekends. It's our opportunity really to show you what we, the genetics program, WashU, Division of Biological Biomedical Sciences, have to offer in terms of research opportunities, also educational opportunities, kind of the class structure, special training opportunities. We're going to go into that not now, but after lunch with the special emphasis pathways and some student groups. And really to also give you an idea of the philosophy and culture of our program. With that, you probably got a taste of it last night with students. We've certainly tried to the best of our ability to maximize and optimize kind of interactions between all of you and all of our students, because they're the best resource in terms of finding out about what it's like being a graduate student at Washington in the genetics program, because they are. Okay, so they're great resources. Definitely ask them questions. More importantly, it's really your opportunity to see what we have to offer in terms of research, didactic, and different training elements, and really to look and see how that meshes with what you're looking for in a graduate program. Okay? So in essence, compare. We're a very good program. I certainly hope all of you have applied to many very good programs. And Tim and I would certainly love to have all of you come here. At the same time, far and away, the most important thing for each of you to do is to make the best and most informed decision on graduate school for you. All of you are different. All of you are looking for different things in graduate school. We want to help you make the best and most informed decision that you possibly can. So having said that, please ask questions now. Ask questions throughout the day. There's dinner at my house tonight. I will not be dressed like this. I will be dressed down. And then you go to the City Museum, where there literally is a 10-story corkscrew slide. It's really fun. But don't wear good clothes to my dinner, because you'll ruin them at the City Museum, which is good. But please ask questions today, tomorrow. If you're like me, on the flight home, you're going to go, I should have asked this question. Email me. All of you should have my email address. Email me next week. Email me in 2 weeks. Email me in a month. Email me in 2 months. That's fine. If you're choosing between our program and another program, it's fine. If you're not choosing between us and you decided 2 other programs are more focused for you, but you want my feedback, kind of a neutral party, email me. I've certainly addressed questions from students in the past who were not really coming here. That's fine. Okay, so anybody's first interview? Okay, a few. Probably first or second for most of you, maybe third. They're perpetually starting earlier and earlier, but at least there's the holiday break, which will probably stop it in January, I hope. So ask questions. If you don't have questions, I'm going to give you 2 questions that you can ask, and you can ask these same questions to absolutely everybody you meet, whether it be students or all the faculty members, and I think it's actually instructive to see what different people say to the same questions. Ask them what they like about, in this case, the genetics program, WashU, St. Louis, the division, and ask them how they would improve it. Don't ask them what they don't like. It's like, oh, I like everything. No, they don't. Okay? Not everyone likes everything about everything. But ask them how they would improve it, because it's not kind of a judgment call. They're like, I want to improve it. So what do I like about the program? Lots of things. I think so with respect to WashU, there are a lot of really top-notch research opportunities in genetics. There's great depth and breadth. I'll go into that. In terms of the program, I think we have a great culture. I think we have great students. I'll talk about the culture / philosophy of our program. I think we've done, this is kind of Tim and me, we've been running it for 10 years. We've really streamlined the educational elements so that they really help students get into the lab and get focused on their research earlier. There's a quote, it's a great quote. I think it's one of the better quotes about grad school, even though there wasn't grad school at the time. It's what we have to do to learn, we learn by, what we have to learn to do, we learn by doing, okay? It's Aristotle. And what that means, more or less, is if you don't know how to do something, you have to do it in order to learn it, which also implies you're going to make lots of mistakes, okay? I think the more mistakes you make in grad school, the more success you're going to have in grad school. There's a fear of making mistakes, and you want to get over that fear of making mistakes in the lab. Because the more mistakes you make in the lab, the more discoveries you're going to make in the lab. Okay? So what we have to learn to do, we learn by doing. So that's kind of our focus on really streamlined training elements. We also do a lot in training in speaking and scientific presentation. You see the video camera up there? Usually it's not videotaping me. Usually it's videotaping students giving talks. Okay? And then we critique the talk in a number of different formats, and the feedback from the students is uniformly positive. And when I ask them, should I keep continuing to take the talks, and they're like, it's painful, but it's really helpful. And it's both, right? It is painful because you look at yourself and you're like, oh my God, I look like a dork. So I'll watch this, I'm gonna go, oh my God, I look like a dork. But it helps to watch yourself talk because there are these nervous tics that you have that you don't realize you have, but when you see them, you're like, oh my God, I shouldn't do that. And whether or not you consciously think about it, you don't do it as much next time because you see it. So I think we're really good at that. How can we improve? We're doing a lot of the stuff the training and speaking we've improved. There are a lot of improvements that we've made. Two areas that I think we can do better in is increased training in basic fundamental statistics, biology becoming more quantitative, essentially in all fields, and also programming. Tim will talk about the genomics class where you get pretty good training in how to program in Python. The criticism we've got in the past 5-6 years from students is, yeah, it's great to learn, but if you don't know anything about Python, it's a pretty steep learning curve at first. So we've done a number of things, but they've all kind of evolved into a kind of 1-credit course in the first semester where Zach Pincus, who most of you will meet today, they basically teach both Python and statistics using Python programming, basic programming skills to do kind of learn basic statistical modules. And the students seem to really like it. And so what it really means functionally for the genetics program students is when you start genomics, you know some Python, which makes the Python computing part of genomics much easier. Some of you are probably pretty fluent in computer programming, and some of you are probably, oh my God, I'm really scared about any computer programming. Okay, so we're trying to lessen the activation energy to learn that. So, and then I think we need to do a little bit better with grant writing training. Tim runs a course in the second semester of the first year where you get explicit grant writing training in terms of a mock grant, but also I think we could have, and there's an NSF grant writing workshop in the first year, but most students submit grants to NIH or American Heart Association. They're bigger grants. We need to have a similar kind of grant writing workshop for that later on. We're working on it. Okay, so that's kind of our perspective of the recruiting week, and it's really your opportunity to see what we have to offer and see how it meshes. I want to go over a quick, and this hopefully will be quick, overview of the weekend and also kind of DBBS structure. Okay, so this was dinner Thursday night. Hopefully you all had a good time. Program overview doing that now. You'll have 3 interviews. They're about 30 minutes or so with faculty members. Lunch with a bunch of students. We'll have discussion after lunch. There'll be kind of brief presentations on student groups and special emphasis pathways. Again, kind of like mine. 2 more interviews. 2 student talks and a happy hour. Dinner at my house, sitting museum. Okay, this is really, really informal. Dress down. Ping pong and darts downstairs. If you want to talk science, stay upstairs. If you want to have fun, downstairs. Tomorrow's pretty abbreviated. We have an apartment and a city tour, so you get to see the city. I think, Tim, you take them to the Danforth campus. So we're at the medical campus. There's Forest Park, which is great, lots of free stuff. There's a zoo, there's a skating rink. I'll be skating there while you're driving by tomorrow. And then 2 miles, there's the Danforth campus, which has the biology department. Another 5 or 6 miles, there's the Danforth Plant Center. I know a number of you are interested in plants, so there's the Danforth Plant Center. Then you have lunch with students, and then hopefully you have safe travels home. Okay, that's the overview. Again, what I said before is we've really tried to optimize and maximize interactions between students here. Please vet whatever I say, whatever Tim says with our students. Okay, they're your best resource. DBBS and the umbrella. Okay, so it's an umbrella. See, umbrella. DBBS, Division of Biological Biomedical Sciences. There are 12 programs. I can't fit. Okay, sorry, there are 12 programs. There are 485 faculty members, 646 students, 459. I'm sure these numbers are not quite right now because they were right last week in other pages. But more or less that's true. A strength about the division is regardless of the program you come into, you're all in the genetics program, you can rotate in and join any of those 485 faculty members. Okay? There are no boundaries to the faculty or the labs into which you can rotate or join. That's a strength. Why is that a strength? Many students, not all, but many students come to graduate school somewhat undifferentiated in your research interests. Your research interests change during your first years, like, oh, I'm really interested in this. And you do a rotation in that, and you're like, well, that was fun. Maybe with this. Oh, this is great. I really like this, but it's quite different from what you thought you were interested in. So this is rare, but I also run the development program. We had a student in development who came in, did some rotations in development programs, and then did a rotation in a lab that does biophysics. Really, protein, DNA, hardcore biophysics. He joined that lab, stayed in the development program. So you can have, it's a very, very flexible in terms of the labs you can join. 12 programs. I'm not listing all of them. Cell bio, micro, plant, comp, there are 11. Then there's the best. The best would be the genetics program. So we don't have to talk about the others for now. The genetics program, I'm going to focus here, if you just look at that, we have 69 PhD students and 9 MD PhD students. I think it's the second largest or third largest program. There are 126 or so genetics program faculty. So these are faculty that are directly associated with it, and frankly about 90 to 95% of our students are in one of these labs of these faculty members, because these are the faculty members that really have a research that really focuses on genetics questions or genomics questions. Having said that, you can still rotate it and join any of the 485. And of these 485, probably about 300 to 320 are really active labs into which students would go. Okay. So that's that. What about research strengths? I'm going to get to this later as well, but I'm going to talk about it now briefly. We have basically 5 broad areas of clear strengths, which span most, if not all, of the key areas of genetics and genomics. So we have model organisms, genetics. I work on flies, Tim works on worms. We also have a lot of people with plants, mice, fish, bacteria, yeast. Genetic basis of a disease, this is when they sequence the human genome. It's the cover of Nature, as you can tell. So a lot of the sequencing of the human genome was done a few blocks that way in the genome center. So we have a very strong contingent and genetic basis of a disease and genomics because, at least partly because of the genomics center, which actually arose from a lab in the genetics department. A strength in epigenetics and clearly a strength in microbiome. This is from Ting Wang's website. Ting, I think a number of you will probably meet, and many of you. He gets really excited when he plays ping pong, so if you see some faculty member dive, microbiome, Jeff Gordon's 2 floors below us. He's really probably the pioneer of studying the microbiome. So we have very strong breadth and depth, and the key point there is if you're interested in genetics and genomics, there are many labs here that you could find in which you will excel. Okay. Program metrics, or key information. Okay. This is what they really want us to talk about briefly. So admissions, 2 interview weekends today, and then in 3 weeks. Okay. When are decisions made? So Steve is actually the faculty. He's in the back helping us out.
Cameraman.
Thanks, Steve. He's the faculty director who oversees admissions for DVBS. When are decisions made? Correct me if I'm wrong. What will happen is we have 2 interview weekends. The admissions committee will probably start making some decisions next week or the week after, but most decisions will be kind of held until after the second interview weekend for obvious reasons. So what I would recommend is if after, like I said, most of you is probably on the early side of the interviews, but if after you've done most of your interviews and you're fairly sure that we're either your top choice or a top choice, it is certainly fine to let us know. You can let me know, you can let Tim know. We'll forward it on to the admissions committee. It cannot hurt. I'm not sure how much it will help, but I can guarantee you it will not hurt. Okay? So 2 admissions weekends, financial support, the stipend is $29,500 as of July 1. Okay, not right now it's $29,000. If you come here, you get a bonus. There's a $5,000 bonus. What is that bonus? If you come here and you get, say, an NSF grant, a grant from the NIH, American Heart Association, any grant that covers 75% or more of your stipend, you get a $5,000 bonus. Okay, so then your stipend goes to $34,500. What you should probably do, you know, depending where you go, $29,500 can go further or less far. There are a bunch of these cost comparisons, or if you go online and do search between like St. Louis, Chicago, St. Louis, Madison, it's cheap in St. Louis. So if you are seriously considering, you just look to see the cost of living differences between the other places you're looking, because $29,500 goes pretty far. Student health is covered, dental and disability travel funds, you get a $600 one-time travel award to go give a talk or a poster. You have to be presented, but go to it. Program metrics, okay. Time to degree is 5.9 years. Since Tim and I took over, it's falling. We've done a lot. I'm happy to talk about this. I'm not going to talk about it now, but we've done a lot to really streamline the graduate educational elements and really help our students get into the lab, working on the research in more depth and more quickly, so that this will probably in the next few years go to about 5.5. Okay, there's 2 talks today, one by Kate Kiefer, one by Sarah DeGenova. Sarah is in her 5th year and she's going to graduate in March. I don't know the day, I should because I'm on a committee. So it'll be about 4.6, 4.7 for her. Completion rate is 92%. That's slightly more rigorous than a retention rate. We're not including students in the 1st, 2nd, 3rd, 4th year here because they haven't. None of those students have completed. This is really groups of students who have come in a class and then have graduated, and everyone in that class has graduated or not. And we have a 92% completion rate. That's high. I think the average is probably, the national average is probably 25%. At the top schools, it's probably between 15 and 20% on average. Publications, what do I mean here? Of our graduates, the PhD graduates, essentially all greater than 95% had at least 1 first author paper. In fact, the average is 2 first author papers, 3.9 publications per graduate student. You know, is more first author publications better than fewer? Yes, but if you have 1 and it's really good, I'm not a big numbers guy. At least 1 is, I think, very important. 1 or more is very important. I had a student, she had 1 paper from her graduate work in my lab, eventually 2, but the second one, she was a second author and it took some time. She had 1 paper from her postdoc lab. She's an assistant professor at the University of Wisconsin. Okay, so she did incredibly well, and those were very good papers. Program outcomes: most go to academia, 60% academia, postdoc, industry, government, other. Yeah, I know there's a typo. It adds to 101%. That's my fault. My dad was a math professor. Anyway, other 50% law, journalism, consulting, so forth and so on. These are just metrics. Okay. That's the key information that we kind of just want to give you. Keys to success in grad school. I think this is probably the important. So what is graduate school about? Here there are no slides, so I'm just going to talk. Okay, this is actually. What's grad school about? It's about research. It's not about classes. It's about discovering something new and different about the natural world of fundamental importance. It's exciting. It's dynamic. It can be frustrating. And you know, when everything comes together, it's really cool. It should be all of those things. Nature doesn't give up its secrets easily, And at the same time, the PhD is the highest academic degree. It should be hard. How do you succeed? I think there are two keys. You're going to laugh. They're going to sound trivial. I think they're closer to profound. Okay, what are the keys? I think you have to work hard and you have to be happy. Okay, see, told you someone would laugh. Work hard, what do I mean? Work hard, think hard, do experiments. Okay, that's your job. Work hard, physically at the bench. Think hard about the experiments, think about what you're doing and why, interact with people broadly, and read. Okay, things will connect. Do experiments. It's experimental science. The more experiments you do, the luckier you get, or something like that. Be happy, what do I mean? Enjoy what you do and where you do it. You want to like the question, and you want to like the people. Because at some time in grad school, experiments aren't going to work. Lines of experiments are going to frustrate you. So you want to be in a position in a lab and in a program where when you get up in the morning, you want to go to work and you want to work with those people even though things aren't working. That is, I think, the real key to success in graduate school. It's not independent, right? It's far easier to work hard, think hard, and do experiments when you're happy. Okay? It sounds trivial, it's not. Okay, so the goal of grad school is to find a lab in which you can excel and then work really hard. How do we help you find a lab in which you can excel? I briefly touched on some of this stuff. There are about 485 labs in the division. You can rotate in and join any of them. Well, Jim, that's a lot of labs. Yes, it is. How do we help you choose those labs? You do 3 rotations. You can do more, but we require everyone to do 3 rotations. They're 8-week rotations. The exact timing is shown there. If you start when most people start in August at orientation, you'll be done by March. You can start as early as June 1. You can do 1 or 2 summer rotations. When you start, you get paid. So once you finish 3, you can join a lab. So we've had some students join in January because they finished 3 rotations by January because they've done a summer rotation. If after 3 rotations you're like, hey Jim, I'm not sure these are I'm not convinced these are the best labs, I want to keep doing a rotation, that's fine. It's far more important to identify a lab in which you can excel than to identify any lab. And that's true here or if you go elsewhere. 450 labs in DBBS, you can rotate in and join anyone. That's a strength. I told you why that's a strength. I'm not going to go over it again. How do you identify relevant labs? There's the website. It's somewhat helpful, but it's still 400 some labs. Jesus Christ, I'm bored going through all these. There's a poster session, which DBBS runs, I think 2 poster sessions during orientation. I run a faculty rotation talk series in August and September, at which somewhere between 50 and 60 faculty members talk. I run it, so it's it's focused, not completely, but somewhat focused on genetics program faculty. It's 3 or 4 faculty a day over lunch, and I buy food for you, so it's a bribe. Students come, bribes work. So briefly, I'm not going to go into detail because I'm taking too long. Like I said, we have a broad and deep representation of faculty in all fields of genetics and genomics, model organism genetics, bacteria, yeast, worms, flies, plants, fish, mice, scores of researchers, and they all overlap. It's not like model organism genetics. People also do genomics. They assess genetic basis of disease, so forth and so on. Genomics, we have scores of researchers. Genetic basis of disease, we probably have hundreds or more, because we're at a very good medical campus. Microbiome, lots of people with the microbiome, 2 really big labs, epigenetics, so forth and so on. Take home, if you're interested in genetics and genomics, we have lots of labs for you, and you can excel in many of them. The rotation system. Okay. I actually don't really like this. What is our philosophy of graduate education? I think this is important, and I'm going to get into this in an unusual way, but that's okay, just bear with me. If I weren't doing what I'm doing now, which is running a number of graduate programs, I also run development, and running a research lab, I'd probably be teaching high school and coaching track. Well, how is that relevant? Let me tell you. I think program directors and faculty members are similar, at least with respect to graduate students. Our job is similar to that of the coach. It's our job to give students the training, the tips, and the confidence and the skill to scale heights that at some point seemed unattainable, too high, too far, and it's our job to do that in as positive, personal, supportive, and critical manner as possible. It's your job to scale the heights. Okay, it's our job to have. I'm going to say it a different way. Many of you will disagree with me at first, so let me explain it. It's our job to help you as little, not as much, but as little as you need to succeed. Okay, what do I mean by that? The goal of grad school, or a goal of grad school, is to become an independent scientist. I think we probably agree with that, right? So when you come into grad school, are you an independent scientist? I don't know if any of you are. I wasn't. Okay? At the end of grad school, was I an independent scientist? Maybe. I'm not sure. But I was a heck of a lot closer to being an independent scientist than I started. Okay? The difference between when you start and when you finish can, at some times, look like a sheer cliff. Okay? When I was in grad school, second or third year, talking to my advisor, we were talking about some research. Can't remember exactly what we were talking about, but he was talking, oh, you could do this, or what about this? But I remember this. I remember thinking, my God, he knows so much, and I know so little. Okay? And the difference to me looked like an absolute sheer cliff, and I'm not exaggerating. Like, am I ever going to get even close to it? And you know, if you go around the sheer cliff, it's not a cliff. What do you see? You see a series of steps. Okay? And the more of those steps that you take on your own, the bigger of those steps that you take on your own, the faster you're going to become an independent scientist. Okay? By on your own, I don't mean in isolation. You're talking, you're interacting with people, you're getting ideas, you're doing all of that, but you're the driver physically and intellectually of your project, and that's important. Now, sometimes you come to a step, and you're like, oh, Jesus, that's a big step. Yeah, sometimes they're big, and sometimes you're just looking at it the wrong way. Then it's our job, your mentors, to really say, hey, you're looking at it wrong. Look at it this way. Oh, it's not as big as I thought. No, it's not. You can take it. You can take it. Sometimes it is too big, then it's our job to break it into. You know, you're going to fall sometimes, get up. You're going to fall, slide down, get up, keep moving. But over 3, 4, 5 years, you can take thousands of steps. And that's important because at the end of grad school, you're going to be at the top of that cliff, or at least a cliff. And that's important because when you look down, what you can say is, wow, I did that. You know why that's important? When you look up, what do you see? More cliffs. Okay, what are those cliffs? Those cliffs are your different kind of career opportunities post-graduate school. But the difference is, when you're starting grad school, you're kind of on a plane, and you look up, all you see is cliffs. You haven't really climbed a cliff with respect to research. So, but now at the end of grad school, you look down and you're like, hey, I climbed that cliff. They're not that big. Which one do I want to do? And again, my advice would be do the one that would make you happiest, because the one that will make you happiest is the one you would do the best job at and bring in the most fulfillment. We're a damn good program. I don't know. You know, they're always damn U.S. News and World Rankings. I don't really care much about it. But we're in the top 5, top 10. There are a handful of programs maybe are higher ranked than us. In terms of establishing a positive, personal, and supportive environment and critical environment that really drives student success and students thrive, none are better. Some might be as good, none are better. Don't believe me? Talk to our students. That's kind of my spiel. I may, if their time permits, give you a few words at the very end. Tim's going to talk about the first year of coursework, and as a transition, I just want to focus what our mission is and have a quiz. The quiz has no bearing on anything. It's to train students really to think critically, write effectively, and speak effectively. Okay, Rodin, Shakespeare, anyone know who that is? She won the Nobel Prize. She was a WashU faculty member. She won it for nerve growth factor and other things. Rita Levi-Montalcini, that's who that is. Okay, but anyway, that's our mission, and the goal is really to train independent scientists, and then below we have, was Reika at dinner? You will meet Reika. She is somewhat unmistakably positive. This is Wendell, guys. Kate's talking tonight at the talks at 3:30. I'll turn it over.
Okay, and so what I'm going to do is talk about the first and the second year, and I'll reiterate some of the same things that Jim's talked about. So the most important thing in your first year is that you choose the appropriate thesis lab, that you find that the projects are really exciting, this is something that you are really interested in, and that the lab environment is something that you're very comfortable with and you feel supported. And there, the most important thing is that you have to feel that you can get along well with the PI. The graduate students and the postdocs come and go, okay, but it's the PI that's really the most important. And the PIs are busy, and so when you're rotating, it's really important that you are sort of very forceful and you interact with the PI, because you really need to make sure that that's somebody who you can get along with the ups and downs with research. So the main thing is that you're going to do your rotations and that you choose a thesis lab. And so this really is a major focus, okay? And so one of the important things that you have to learn in graduate school, if you haven't learned it before, is time management. juggling between your rotations and your coursework and other activities that you might be doing. And getting these skills, these aren't really skills we can teach you. You can certainly come talk to us and we can give you hints about time management, but this is actually something that largely you're going to have to figure out yourself. But these skills that you learn, you'll use them for the rest of your life. Okay? So you'll do your rotations, and as Jim mentioned, most students will choose a lab after 3 rotations. But again, we are very flexible. If you feel that you really want to explore and try out some other labs, we're very amenable to that. Okay? So then coursework. Okay, and so the figuring out what your thesis lab is the most important thing. The courses are less important, but they still are important. And the bottom line is you need to get a B average to maintain your standing in graduate school. Okay, and so in the fall you take most students will take molecular cell biology and nucleic acids. And these are sort of advanced versions of the courses that some of you may have already taken as undergrads. And here they'll concentrate much more on the primary literature than textbooks.
Nucleic acids, it's a good class. It sounds really boring. It's basically gene regulation.
And so, Jim also mentioned there's this fundamental in biostats course, and it's a short course, I think 6 to 8 weeks, and it has a Python programming component to it, which will help you for when you go to the genomics course later. As Jim mentioned, in terms of trying to figure out what labs to rotate in, the faculty talks, but also in September we have a retreat, the genetics program, and we do it also with the comp systems biology program. And so there will be young faculty there talking about their labs, plus you'll be able to meet other faculty who are attending and other students, and so you may get more information on possible labs that you might be interested in rotating in. Okay, so then in the spring...
So one quick thing. So we often have students who come with a pretty good background in those two and are really interested in kind of the computational side. There is a computational molecular biology class that runs in the fall. Talk to us, you can petition us to take that class. In general, if you're interested in that and don't have a very strong background in it, take genomics, which we'll talk about now, and then do computational molecular biology in the fall of your second year. Sorry.
Okay, so then in the spring are really the courses. So actually, one more thing. If you're struggling with your courses, and occasionally a student does, this is something where you want to come talk to Jim and I, and we can arrange for tutors to help out. This is not a problem, and just don't be shy about it. That's true here if you feel that you're struggling in spring semester courses. Come talk to us. Okay, these are really the core courses for the molecular genetics and genomics program, and that's the genomics course and the advanced genetics course. And what we really are doing here are giving you, in these courses, skills that you will be using as an independent scientist. Okay, so in the genomics course, of course, it's going to go over all sorts of aspects of genomics. But a key part of genomics and a key part of all biological science is now very large data sets. Okay, there are often you'll come across RNA-seq data sets with 2 million, 20 million data entries, proteomics data sets, lots of things, lots of high throughput data sets. And you need to be able to not be afraid of them, but to be able to mine them and get whatever information out of them you want to be able to do. And this will greatly empower you both in your thesis lab and later in your career. And so the computational lab that they have really teaches you how to do this. So you'll learn the Python scripting skills so you can parse the data into various multiple parameters, so you can look for an RNA expression level that depends upon 10 factors, and then you can sort them and pull them out rapidly. So this information really will empower you as a skill
but it also will allow you to then think about genomic level experiments and how you would use that to essentially obtain data for your thesis, okay? So in the advanced genetics course, this does two things. First, it discusses in great deal how you use genetics to dissect biological processes, how you use forward genetics, reverse genetics, and the logic of genetics to dissect biology. And this really has been a key part of biomedical advances for the last 30 years. So the second part of the course is to write a research proposal, okay? So a key part of being an independent scientist is to be able to come up with a hypothesis and have experiments and design experiments to test that hypothesis. So this proposal is a little bit different than a grant writing proposal because what we want to do is hone your skills to come up with a hypothesis and come up with experiments to test the hypothesis. And where do hypotheses come from? So this comes from knowledge of a field and coming up with questions related to gaps in that knowledge of the field. Where do the tests of that hypothesis come from? Well, approaches that you learn in genomics and approaches that you learn in advanced genetics. So here the goal of this proposal is really to try to get you to hone your ability to devise a hypothesis and devise a series of experiments to test the hypothesis. So at the end of the semester in May, we have an in-class prelim, and it's greater than 90% of it based on the genomics and the advanced genetics class, and about 5 to 10% on molecular cell biology and nucleic acids. Okay, then the next thing that's related to the program. So one thing I didn't mention about the research proposal, this research proposal should be some new topic area that you haven't encountered in your previous research experience. Okay, so the in-class prelim is just an in-class prelim where essentially there are questions that you answer, the vast majority of our students pass the prelim because we prepare you well here, and tell you these are the kinds of things that you need to know for the prelim.
We haven't had a student fail in 5 years, partly because they have the last 10 years of prelim, so they're also similar questions.
Okay, so after this, for those students who have joined the thesis lab in March or April, what we have is a written prelim, and the ideal deadline is to have it done by October 1. And this prelim is on your thesis topic. And basically, 2/3 to 3/4 of that prelim, written prelim document, is a review of the field for your thesis topic. And so this gives you an opportunity to really delve into the literature of your thesis topic and really know the literature and what are the questions based on the classical concepts that came up. And this really gives you a chance to go back and read papers from the 60s or 50s that really were foundational for the field, okay? And you can then discuss them as part of the background for your proposal. And then there's usually one aim, experimental aim, of an experiment that you're proposing to do in your thesis lab, but it's not a big major issue if some data comes along that says that aim is not going to be feasible at a later date. So the goal here really is to really immerse yourself in your thesis topic so that you're really ready to charge ahead, okay? And then most students we want to, within no more than a year, for you to do your thesis proposal, and ideally less, okay? Because the thing to remember about the thesis proposal is this is where you're now going out and using other faculty member to provide advice on where you think you're going with your thesis. And the longer you wait to get this advice, is essentially detrimental to you. You want to get this advice sooner rather than later. So in the second year, all students will TA for one semester, either in the spring or this fall. and all students take an ethics course in the spring.
Wherever you go, you take ethics. It's mandated by said government.
Okay, and then, essentially, your contact with the program, becomes greatly diminished once you are in a thesis lab. We certainly expect that you will attend the retreat and present your research at the retreats. And most students, attend the retreat except for when they're sort of getting ready to write up their proposals and they want to just focus on moving on. And so, of course, what's that?
It's just a lot of fun.
Yeah, it's a lot of fun. And so, of course, we expect you to publish your work and present your work. We want you to present your work at WashU, certainly at the retreat, but there actually are many other forums for you to present your work and to get comments back. And of course, then you will graduate in 5 or 6 years. And later on, we'll talk at noon about special emphasis pathways and other kinds of activities that are available. So, questions? Yes?
Sorry, this is a question about the bonus you talked about. So if you're on a NSF grant or something, is it just the years you're using it, or how does that work?
It's definitely the years you're using it, Steve, do you know?
It's the duration of the...
It's the duration of the grant.
Okay.
That's changed somewhat in the past few years. Okay.